The next plenary talk in this session is by Professor Satoshi Kawata from Osaka University, which I believe is called tip-enhanced Raman microscopy.
Thanks, Nikolai, for your introduction, and I'd like to thank organizers for inviting me here. Actually, Ichon is not far from Osaka. It doesn't take more than 2 hours by flight, 1 hour 45 minutes from Osaka. So it's more like a domestic for me, but I haven't been this area. I always stop by airport, so I was very amazed to see the development of the city in Incheon. So my great pleasure to be here. And the title, today's talk is more like a microscopy. And differently from the previous two talks, the spectral range for the Raman spectroscopy should be wide, broad. So it's not the like a lasing. We use a laser for excitation, but the scattering should be wide in spectral range. That is the difference. And this is my main research topic. So while we do a lot of plasmonics or nano studies. The outline of my talk is first I would like to introduce you what or why tip-enhanced Raman scattering microscopy is. And then I will talk about the resolution exceeding beyond the limit of not diffraction, but the limit of the plasmonics. And number three, I would like to talk you about our recent development of the three-dimensional imaging with using a metallic actual particle in a living cell as a microscope. And number four is one of the latest research progresses, that is deep UV tip-enhanced Raman microscopy and deep UV plasmonics. So as everybody here knows well, the spatial resolution of the microscope is limited by the diffraction of the light, which is comparable to the wavelengths or half wavelengths of the light. And to have a high resolution, people want to use a short wavelengths of the light, like an X-ray or an E-beam. But we don't want to change the frequency. If you want to see the sample in red, you don't want to use UV. You want to use the red light. So the frequency is given, omega is given, but you want to have a high spatial resolution, short wavelengths. And that can be possible by controlling the light speed c. And if you can reduce the speed of the light, you can have a high spatial resolution. This is used commonly, traditionally, in optical microscope. People put your sample in water, in a high index oil to have a high resolution. This is typical. Or you can put your sample, if it is possible, in glass. But the point here is rather than using those dielectrics, if you want to have a much higher spatial resolution, you may want to put your sample in metal. And if it is possible, if you put your sample in metal, in the metal there is a light that is associated with a plasma wave. And since the electrons collectively oscillate, this is associated with the light, photons, electromagnetic waves. Although this immediately disappears in the metal. But the wavelength is much shorter. At the resonance, it is 0. And of course, it is not possible to put your sample in metal, but you have a light in metal. And then you may naturally think that you can put your sample on the boundary between the metal and the air, or whatever water. Then you have a surface mode of the plasma waves. Then this is still can be shorter than the wavelengths of the light in air. So on the metal surface, if you can put on your sample on the metal surface, you can have a microscope with a high spatial resolution. So the light speed reduces and wavelength shortens. And of course, this is also doesn't have a long lifetime because light is absorbed by the metal, but still much better than this condition. You can get a reasonable high resolution optical microscope image. So this is a fundamental concept of us, and you have to put your sample near field of your metal surface. So this is near field optics using the surface plasmon polarizones. And you can do this one on the metal surface, but if your metal has sample has a distribution, if you want to get an image, even though if you can have a high resolution, there is no way to detect the image with a high resolution. So we, rather than using a metal surface, we propose to use a metal tip which scans on the sample surface. So the plasmon is no more propagating plasmons, but a standing wave at the end of the particle, tip. And I don't want to discuss the detail of this one, but this is the concept of the aperture scanning optical microscope. And we have done it, and the mechanism, the method to get an image is simple. Just you scan this metal tip along your sample surface, and it forms an image. And this was proposed many years ago, more than 20 years ago. We proposed to use a metallic probe tip for near-field scanning optical microscope. We use an STM tip to scan on the sample surface, and the photon is confined at the end of the tip if you give a light from a side or from the bottom. And so plasmonics can be well used for near-field optical nano-imaging, but besides, it has a feature advantage, which is an enhancement of the signal. The resonance of the surface plasmons as a standing wave or a cavity, in the other word, you can have an enhancement of the signal. So besides the nano resolution, you can have a signal enough. In particular, Raman scattering is an extremely weak photon process. The cross-section, scattering cross-section is 10 minus 30 inverse squared centimeter. So it's not really possible to get an image, signal, except Rayleigh scattering from your molecule in an ordinary sense. You need to take 3 hours, 1 day, 2 days to get an image usually. But if you have a metallic tip, it enhances the scattering field. We don't want to enhance the excitation field. We want to enhance the scattering field. So even for Raman scattering, in this case we used an AFM cantilever, and the system was GAF mode. We can get an image and or spectrum as well. So this is one of the very early experimental results of us. We have an adenine molecules as nanocrystals, and you gave a light from the bottom, and the tip comes to the sample. And if the tip comes to a molecule, you have a signal enhancement in Raman spectrum. And if the probe is far, the signal disappears. And if you see the factor of the enhancement, it could be easily like 1,000 to 2,700. It depends on the vibration mode. But you can have an enhancement of the signal besides a high resolution. And I show you just 2 images today as examples how tip-enhanced Raman works well. And this is an optical microscope image of carbon nanotubes. And since this is a conventional optical microscope, the resolution is limited by the diffraction. And so this is 100 nanometer. So the spot size is about this size, 100, 200, so about this size. So you don't see the details. And also this is an optical microscope, but we had a filter to pass only the G-band vibration mode of the carbon nanotubes. The intensity is only contrast only the information from the carbon nanotubes. And since this Raman signal is very weak, so the image is noisy. But still you can get the information of the carbon nanotube G-band vibration mode with an optical microscope. But this is a conventional optical microscope. And but if you have a metallic tip, plasmonic tip, and if you scan this plasmonic tip on the sample surface, what you can get is this one. So here, due to the diffraction limit, you may think that there are only 2 carbon nanotubes distributed, one here and one here. But by using the tip diameter was, in this case, 20 nanometer. So the resolution is about 20 nanometer. And you could see the details of the distribution of the carbon nanotubes in this area as well as this area. And also, you don't see any other molecules or catalysts except G-band vibration mode, because this is a spectroscopical imaging. So this shows the metallic tip works to get a high spatial resolution. The other example here is the same as previous one. The sample is carbon nanotube, and this is a topography, AFM image of the carbon nanotube. Here the carbon nanotube is formed as the letters C and T. And this is topography. This is optical microscope image filtered at the G-band. And pixel size is 10 by 10 nanometer. And total frame size is 390 by 680. So this is about the size of the light spot you shine. And in the light spot, you scan the tip to get a high spatial resolution. And here, what we wanted to show you was another image of the tip-enhanced Raman scattering microscope image, which represents again G-band, but not G-band intensity, G-band spectral peak shift. So the peak position varies position by position. So the peak of the G-band vibration mode varies by position. And this means that the carbon nanotube, this one, is distorted locally. And why this is, let me show you. Maybe this one. Yeah. So if you have a torsional or tensile stress, the tube lattice is deformed and the spectral peak position changes. So why we have a shift variation in image? Because we push-pull this carbon nanotube by AFM tip on the substrate. So locally you have a strain in carbon nanotube, one single carbon nanotube. So you have a distribution of the distortion, lattice constant variation in one carbon nanotube. So this shows more precise demonstration. You have one linear carbon nanotube, and the vertical axis is frequency, lateral axis is position. And if you don't deform the carbon nanotube, the peak is a constant. But if you push the carbon nanotubes locally, you have a peak shift, further peak shift from this position at A, B, and C. So this tells you how the carbon nanotube is deformed inside. So the next one is about the resolution. And the resolution in ordinary tip-enhanced Raman scope is typically 10 nanometer to 20 nanometer. It is not possible to sharpen the like a silver tip, like a 1 nanometer. This is metal. Metal tends to be round by metallic bonds. And secondly, your imaginary part of the dielectric constant is not negligible. So you have some amount of the imaginary part to have a broadening. And also you need some amount of the atoms to oscillate the plasmons, electrons. So typically, unless you do anything in particular, the size is 10 to 20 nanometer. And in this talk, I don't talk about the gap mode, which was talking in the previous two speakers, because the sample is distributed and the samples are dielectric, so we can't bring the other tip. So this is a typical ordinary case of the microscope. But there is a way to improve the resolution drastically. And so this is a light spot under the tip, which is typically like a 10 to 15 nanometer. But in this light spot, we gave a pressure to deform the molecule, and deforming is made by one. So if you follow the change in Raman spectrum, you can see the molecules with a higher resolution. And we have done this experiment, and this is carbon nanotubes, and we gave a tip with some pressure, and 2.4 is maximum, otherwise the carbon nanotube becomes plastically deformed. And then you see the peaks of the G band can be separated by G plus and G minus. G plus is a vertical, longitudinal vibration, and G minus is an azimuthal. And if you see the intensity changes and also peak changes, and I don't want to discuss much, but this azimuthal vibration makes a spectral change because the diameter changes and also intensity changes. And we do this experiment to get an image, and if you scan the tip crossing the carbon nanotube, you have a resolution like 4 nanometer. And the vertical axis is not intensity, but the peak shift. So this is not a linear system, but very non-linear system. So the real resolution is limit is nearly 1 nanometer. Do I still have time, right? Yeah. And number 3, 3D imaging. And we have shown this kind of result to biologists, and but the people complained us. Our tip can get a high resolution image of only the membrane surface, not the inside. But their interest is not only the membrane, but also inside the cell. So rather than using the tip, we use a small metallic particle which can move inside the cell. And we have done many methods, including laser trapping to scan the cell with a particle. But cell does not allow us to scan herself. The particle can move only the area which allowed it by the cell. It's a cell function. So we gave up to force to scan the particle. We leave the particle move as they like. And then what we have done is particle use of the particle. We have a dynamic imaging of the cellular transport pathway, and here is a trajectory of the particle, metallic particle in a cell, and this looks random, but sometimes not random. And during the motion of the particle traveling in the cell, this particle keep enhancing the Raman scattering at the molecules near to this particle. So I do it again. So let me see. Yes, particle is moving randomly, and you have a Raman spectrum simultaneously. And this position is precisely detected, and here it is driven to this place, and it is traps. So the spatial resolution is not given by the diffraction. Spatial resolution is given by the position-sensitive detector to find the right position of this one metallic particle. And I don't show the details in this paper, but so far as you follow one particle, not two particles, you can have a resolution much finer than the wavelengths of the light. You can just find the peak position of the point spread. And simultaneously, you can detect the Raman spectrum. And the mechanism in this previous experiment was follows. First, a particle is taken up the cell. Cell wants to take up things from outside. This is called endocytosis. And then once this is taken, this is first Brownian moving as a Brownian motion randomly, as you saw. And during this time, the spectrum is random, changing time by time, position by position, because the particle meets many molecules. But when it comes onto this conveyor to transport by kinesin and dynesin, like this motion, this sees only the molecules here, so the spectrum becomes quite stationary during this process. And then it is taken up to the lithosome. Lithosome works to digest the materials like a virus. Again, the spectrum becomes stationary. So by following the by following the motion of the metallic particle, you can get the 3D image of cell pathway, and this is 3D representation of this 3D imaging, and these different colors tell you the different molecules at the position where particle stay and move. Like a CN stretching CH23, and this is I can't read. Tryptophan, yeah. And in the end, I hope I have a time to talk a bit about our latest work, which is deep UV tip enhanced Raman. And it is nice to do the Raman spectroscopy, Raman microscopy with visible light. But if you use a deep UV, the information Raman spectrum you obtain is completely different from that with a visible light. Raman spectrum is just a spectral shift from the Rayleigh scattering. So whatever you use the wavelengths, basically the spectrum won't change stationary. But in deep UV, the spectrum changes a lot from this one. And this is because of it is a resonant Raman. You have an electron transition and then scattering. So this is very sensitive and so very much efficient to emit the Raman spectrum, Raman signal. And the information what you see is different from this one. This is basically like a guanine adenine, DNA nucleotide bases, and tryptophan, and tyrosine, proteins, aromatic acids, where invisible you see only lipids and some of the cytochrome Cs. So we wanted to use deep UV to see the real biomolecules, nucleotides and aromatic acid molecules with use of an UV light. And you don't need a high power of the laser because the scattering power is much higher than visible if you use deep UV. And the resolution is naturally also higher due to the short wavelengths. And the resonance, it is on resonance, so the intensity of the excitation is much, much weaker. And this is a report by the chemists, and they have shown that the adenine, guanine, cytosine, thymine show very different Raman spectra by using the UV, deep UV lights. And these are basically second harmonic of an argon, a multi-line argon ion lasers, so they are available. However, the problem is still, even if you don't use high power of the laser, still the problem is photo damage. It is resonance to Raman, absorption. Similarly to fluorescence, the light is absorbed by the molecules. Fluorescence case absorbed by fluorescing molecules. But anyway, photo damage. So we have discussed the issue of photo damage, and we have seen how molecules are broken, showing, giving the different intermediate molecules and how they disappear. But the main point is not to study the photo damage, but to overcome the issue of photo damage for deep UV Raman spectroscopy. And in the end, recently we have found that if we put your sample in lanthanide ion solution, molecules are not burned. Molecules do not provide reactive oxygen species. But lanthanide ions, the energy is transferred to lanthanide ions that emit fluorescence. So the energy is not consumed as a thermal effect, heat up, but luminous from the lanthanide ions. And this fluorescence is very far from the in the range of the spectrum, far from the Raman scattering range. So you can easily separate them. And we have done many experiments. And here, this is a conventional optical microscope image. And this is the, so this is morphology. And this is an image of the cells before the UV laser beam scanning. After UV laser scanning, the molecules, cells are gone. But if you have like a terbium, lanthanide ion, the molecules remain morphologically, europium, and this one also. So this is particularly purposely given a high power of the laser, but still the molecules are not burnt. And not only the morphology, but also the molecules themselves remain after the experiment. And this is an guanine and adenine distribution image of an HeLa cell after the experiment with terbium ion solution. And this is the spectrum of one position. So we could see them, and we are working on applying plasmonic type study with this resonant Raman spectroscopy. And plasmonic materials are basically, plasmonics is basically visible near infrared, near UV range. So far as use silver or gold. And gold, silver become no more plasmonic metallic. In deep UV, it become dielectric. And but there are some good candidates. For example, if you use an aluminum, it has a resonance of a localized surface plasmon is at 140, so it's very plasmonic in deep UV range, and imaginary part of the dielectric constant of the aluminum is small enough, while it is so large, invisible. And the indium also is a good candidate, and they have an oxidized as a layer with a few nanometers, but this is still much better than the oxidization of the silver. The thickness of the oxidization of the silver is much thicker. And there are other candidates, but those have a high, large value for the imaginary part, or not much short enough, or flammable, or for the deposition temperature is too low, melting temperature is too low. So we are working for making the good tips with aluminum, and we use an aluminum for tip enhanced Raman, and this is just one of the demonstrations we published many years ago. By having the aluminum tip, if the aluminum tip is near to the adenine, you have an enhancement of the signal, as see, and if the tip is far, this is gone, and this is crystal violet case again. So we are working for combining these ideas as a deep UV tip enhanced Raman scattering microscope using the plasmonic metallic tip. And this is probably the last view graph of mine. What I have taken here is published a few months ago, or 1 month ago, in Chemical Review. And people may think that if you have a high resolution, the spot size is too small. So, but actually, and the signal may become small, but this is not the trade-off. If the resolution is high, the enhancement is high. And to satisfy this one, I propose to use 3 concepts. One, deep UV resonance, and second, I don't read these letters. Second, plasmonic resonance, and the number 3, nonlinear effect, like a Cars SARS, Cars SRS. And we have worked on combining those 2 or 3 for having the high resolution and high enhancement. As a summary, I have talked about Raman scattering microscopy, and it is now ready to use. And even line confocal works well, and plasmonic deep UVs and nonlinear, as I mentioned in the previous view graph. And the resolution is typically 10 nanometer, but using different mechanisms, it can be higher. And the enhancement is for our applications, like 1,000 to 10,000. It is because we are not interested in one particular wavelength of the light, which is different from the laser application. We need a range. We want to enhance all the vibration modes so that we can't claim too much high resolution. But sometimes people claim a lot. And reproducibility is so important. We have a publication last year. And even non-scanning, high resolution, super resolution imaging based on plasmonics is important. But we don't do much after we published one paper in Nature Photonics. And 3D imaging and deep UV. So those works are done with my students and my postdocs and my colleagues. And works are funded, supported by the funding agencies. So thank you very much for your attention.
Thank you very much. We have time for questions, please. Or maybe I can start again. So Satoshi, you started with this premise that what we can control is the speed of light, and this is argumentation for plasmonics. So we know now very well that in the infrared part of the spectrum in two-dimensional materials, we can have a very considerable confinement of plasmons and polaritons, exceeding 100 if we talk about, say, 10 microns wavelengths. So my point is there any reason, any point trying to do spectroscopy with high resolution in the infrared? From perspective of biology and chemistry, not from technological physical aspect, but from the beneficiaries of the spectroscopy?
Very good question. Infrared is also an important range for microscopists, and differently from Raman, the cross-section is very high, and since cross-section is too high, they don't use much infrared. But wavelength issue, yes, we can play with reducing the speed or plasmonic method using like graphene or other materials for super resolution microscopy. And I don't know how much. I am not doing IR this moment, but I don't know how much people are working on. And there are some works on infrared plasmonic absorption microscopy, like Fritz Kalman and others, but they do not use they use still metals, not the graphenes and those kinds, and they measure they use an interferometric technique. So the answer is I don't know.
Yeah. And then another question. So you talk about this entry into the UV part of the spectrum and use some protein Raman spectra as an example. I'm just wondering, sort of, if you go to 200, 250 nanometers, you enter the wavelength in which protein starts to denaturate with light, right? So is there any problem in spectroscopy when you start sort of using wavelengths at which the molecules become to change, like the denaturation of proteins? Is it a problem going to from the point of view of biology, is it a problem going to
There are particular wavelengths fitting to the electronic transition for individual proteins, individual nucleotides. Those are at like 244, 257, 228. So we don't need to go further in those range. And if you have a skill, better to use different wavelengths for the different molecules. So besides Raman scattering spectroscopy, you have an absorption spectroscopy. And you can play with those. And there is a wavelength for each.
I see. I'm happy to take another question. Yes, please, over there in this row. Yeah. Can you bring microphone to row 7, please?
Thank you. I very much like the cell uptake experiments. Are there ways to internalize maybe a dimer to get even a better field enhancement? So if you internalize a gold nanoparticle dimer, then you have the field enhancement in between, and you would probably get a much better signal.
Yes, dimer or a gap mode gives a higher enhancement. But if your sample is distributed, how do you bring these dimers in the position where you are interested in? Is an issue. So practically for the microscope application, the gap is not behind the dimers, in between the dimers. And the sample may not be inside, in between, can be below 2. So practically it won't work for most of applications. But if you want to see like the one molecule, as the first speaker talked, you can bring your molecule in two particles, then you can have a higher enhancement. And also, if it is too much high in enhancement, in spectroscopy, it is in a very high Q. So for Raman spectroscopy, you want to know the different vibration modes in one. So if one mode is seen, the other mode disappears. So I don't claim too much the factor of the enhancement. 10 to 3 is, to me, reasonable.
Thank you very much. I think it's time to thank all the speakers today, and in particular Satoshi Kawata for the last talk. Thank you very much.